Okay, so thank you very much for the invitation. I recently joined the Cochin-Institut in Paris, and I'm really excited to join the community. So today I'm going to talk to you about preclinical models of myeloid malignancies, which is what we use in the lab for studying the interaction between the leukemia and the microenvironment. So, as you know very well, the hematopoietic system is a highly structured hierarchy organized in very immature progenitors that reside at the top of the hierarchy and that have the ability to differentiate toward different lineages and generate all the blood cells that are in an organism. So when mutation occurs in this compartment, there is the generation of leukemic clones that are clearly aberrant and are not able anymore to differentiate toward specific lineages, and they accumulate in the organism, taking the space of the normal hematopoiesis. From a genetic point of view, the mutations that cause this overgrowth are starting to be known, and they are known since a while, but now we know very well how do they classify into subgroups and how do they influence the clinical behavior of the patients. However, even though this heterogeneity starts to be dissected at the molecular level, still this hasn't translated yet into novel treatment strategies. So the treatment protocols for these patients remain the standard chemotherapy that are administered both at the induction and the consolidation phase in most of the cases, and they are quite toxic for the patients, and also they can generate the occurrence of resistance within the leukemia compartment. So with few exceptions, there is still a lot of room for discovering novel potential targets for helping the behavior of the treatment and the life of the patients. So one possibility is looking at the bone marrow microenvironment. So I like to describe the environment as a first shield of protection of the hematopoietic system from any kind of insults that come from outside. For example, inflammation or infections or leukemia itself, which is an insult for the hematopoietic system. So the microenvironment I am interested in is the stromal microenvironment, which is a very heterogeneous tissue constituted by different kinds of stromal cells that contribute to the tissue homeostasis. So we can describe a very important vascular component, which is lined by specific endothelial cells that define specialized vessels. The mesenchymal component is also very abundant and fundamental for the tissue development and homeostasis. And the mesenchymal cells can be described as either a very immature compartment of progenitors and stem cells, or different lineages of committed cells. Indeed, these cells are able to generate osteoprogenitors and mature osteoblasts lining the bones, but they can also generate chondrocytes or adipocytes, as well as pericytes associated to the vessels. The bone marrow is also an innervated tissue with specific stromal cells lining those nerve fibers. And I can go on and describe many of other cell types that have been discovered recently. And all of these stromal cell types that reside in the bone marrow have been largely studied in the hematopoietic field for their function of support of the normal hematopoietic stem cell function. So why do we study the niche also in the case of leukemia? So we have understood, like a few years ago now, that not only hematopoietic stem cells but also leukemic stem cells require the niche to be able to exert their stem cell function. So like homing to the tissue, for instance, and finding nutrients, as well as resist to chemotherapy. So this concept has evolved in the years with the understanding that not only the leukemic stem cells do require the same niche as the HSCs, but they do also modify this niche and transform it into a leukemia busting unit, and is not anymore supportive for the normal hematopoietic stem cell function. So a more novel concept that has arisen mostly from genetic mouse models experiments has shown that not only the niche can support the leukemia, but the modified niche can also be the place where a leukemia arises. So modified niche derived from inflammation or aging, chemotherapy, can be the environment when there is occurrence of mutations within stem cells that are stressed and they become leukemia initiating cells. So to try and study the niche in myeloid malignancies, we could use patient samples as something we do, and it's really useful. However, patient samples are not enough to try and answer all the questions that we have to understand these diseases. So we need to use preclinical models, and so I'm going to describe a few of them, especially the mouse in vivo ones. So the one that is the more classical preclinical model that is used in leukemia is PDX, patient-derived xenografts. So this consists in inoculating leukemic cells from patients directly into the blood system of a mouse, immunodeficient mouse, and this is recapitulating a full-blown disease, human disease in the mouse. So clearly creates a chimeric environment, and this is one of the downside of the model. However, one of the upside is the fact that the mouse system is very well characterized, and so we can use several tools and genetic tools, fluorescent tools, to try and study the interaction between leukemia and the niche. Other models that have been discovered, invented, I would say, introduced recently, are in vivo organoids, which are I'm gonna talk a little bit at the end about these models. Many different labs have developed these small tissues that can recapitulate the bone marrow niche and support acute myeloid leukemia growth. And one model that can be used, avoiding completely the use of animals and still being a preclinical model, is having 3D in vitro organoids or organ-on-chip. So regarding the first model, which is the one that is mostly used in the community, I'm going to show you some data that we produced during the last years in Dominique Bonnet's laboratory at the Francis Crick Institute that we are wrapping up now for publication. So to try and explore the molecular alteration of the leukemic bone marrow environment, we have used immunodeficient mouse models carrying fluorescent reporters in specific niche components, and we have engrafted them with the AML patient samples. So with the aim of isolating the stroma and study their transcriptomic signature. So the very first picture that we had up on this study of this transcriptomic data was looking at the normal stromal cells in the absence of leukemia and see how we can better characterize them by using their transcriptomic signature. So we could isolate and identify 8 functional clusters within the stromal cells that define specific niche cell identity. Some of these clusters are kind of known already. For example, the cluster 2 is called endothelial cluster, because it accounts for genes that are involved in processes such as angiogenesis, tube morphogenesis, vascular development, etc. And it's clearly a number of genes that is highly expressed in endothelial cells. So all the other clusters, some of others are really interesting, and they account for novel cross-relationship between stromal components. So this is a map summarizing all these interactions, and it's a way of better defining how the niche function in homeostasis. So then we look at those clusters in the presence of patients, leukemia patients, and so what were the major changes in the presence of leukemic engraftment. So as you can see, some of the clusters were unchanged, while the others were dramatically downregulated, so meaning that the function of those genes is changed in the presence of leukemia. So we could identify an interesting downregulation of the skeletal function within bone development, specifically in the nestin low and the osteoprogenitor cells, while some of these genes were, on the contrary, upregulated in cells that are defined as a pericyte, as it is characterized so far. So it's very interesting to see that the niche can be plastic and react to the presence of an insult by switching from a function to another. We also observed the strong reduction of genes and the clusters involved in muscular contraction and vascular tone, and this is very interesting because it can be associated to a vascular insult that has been shown previously in the field, so I'm going to come back on that later on. We also observed a downregulation specifically in the endothelial and osteo lineage cells of genes related to the innate immunity regulation. So all these data, which are it's very wide picture, I'm just giving you some hints of the data, are a very good way of exploring the molecular alteration induced by human AML in the bone marrow niche. So what I've been showing you so far is a picture of the niche with very high leukemic engraftment. But what we can do with preclinical model is also looking at the leukemia while it is developing, which is something, unfortunately, we cannot do in patients in most of the cases. So we could take a picture of the niche, in particular the mesenchymal niche, at early and late time points by using a tool that has been recently published by our collaborators at the CRIC and used in lung metastasis projects. So this is a painting tool that allows the leukemic cells to give a fluorescent proteins to the cell that are surrounding them. So these cells are painted, as you can see in the image here, and we can isolate them while they are still alive and study their transcriptomic signature. So that's what we did, and trying to identifying what were the genes and the pathway and processes that were active already in the niche at early time point in the leukemia development. Now we are trying and identify what among these are interesting candidate genes that are essential for the establishment of the leukemia niche crosstalk. Okay, switching now to the vascular microenvironment. So it's a very interesting tissue, as I mentioned briefly in the introduction. So it is interesting because since ever, I would say, we know that the vasculature is essential for bringing oxygen and nutrient to the tissue, as well as favoring the cellular and metabolite exchanges. However, recently, I would say in the last 10 years, we have started to understand how the vascular microenvironment in the bone marrow is organized and structured in terms of specific markers expressed by endothelial cells and in terms of functional vessels. So now we know that endothelial cells in the bone marrow are also essential for supporting HSC's maintenance. So what we know in leukemia is that the vascular microenvironment is essential for supporting the T-cell leukemia growth and homing as well as survival by producing fundamental chemokines for their support. But we also know that the vasculature is participating to pathogenesis of myeloid malignancies and is a sanctuary of chemoprotection in the case of CML and AML. So we recently discovered that the vascular microenvironment in the presence of leukemia is not only important for supporting the leukemia, but is on the other way around disrupted in a way by the presence of the leukemic cells. So this is a paper we published a few years ago now, and is in paper in which we have studied the vascular microenvironment in PDX models by using intravital two-photon imaging. And this allowed us to visualize the increased permeability phenotype that we observed in the presence of leukemia, a phenotype associated to a loss of functional sinusoidal vessels and an increase of hypoxia in the bone marrow tissue. So by looking at what was causing this damage in the bone marrow, we identified by sequencing one potential target, which is nitric oxide, that we found upregulated in the bone marrow tissue and higher produced by endothelial cells. So we are now trying to translate those data to patients, since, as I described before, these PDX models are chimeric environments. So we would like to see whether this vascular phenotype are also present in patients. So to do that, unfortunately, we cannot image with microscopy patients, but we can image them with diagnostic tools. And so we have teamed up with specialists in magnetic resonance imaging and clinicians, and we set up tools, the imaging tools for visualizing the bone marrow vasculature in the PDX model. So as you can see from these images here, we could visualize the bone as well as the bone marrow, which is the white part in this picture here, and we could see what were the vessels feeding the bone marrow. So by using dynamic contrast enhancement MRI technique, we could visualize with a dynamic protocol the increase in contrast enhancement in the bone marrow and then the decrease of it. And these parameters can be linked to the ability of leukemic of vessels to bring a nutrient and anything else to the tissue, and can be linked to different other vascular phenotypes. For instance, we could extract parameters linked to blood volume and perfusion, as well as vascular permeability and blood flow. So by analyzing control animals first, we could see that already in normal aging process, there is a detriment of the vascular function. We could observe reduction of perfusion and increase of vascular permeability. So we then apply those parameters to cohorts of AML PDX, and we could observe that all these parameters were affected in the presence of leukemia. Here we can see the increase in vascular permeability with different patient samples. And not only we can visualize the damage to the tissue as an average value in the bone marrow, but we can also have a map, a 3D map, sorry, a 2D map in this case, but we can go further. But for now it's a 2D map of the bone marrow tissue, in which we can see the intensity values related to the vascular parameters. So for instance, we can see that there are areas that are very poorly perfused in the presence of leukemia, and at the same time this same area are highly leaky, which means that vessels are not able to retain their normal flow, and so this is the reason why the liquid is just released everywhere randomly. So this is like a patient in which we could observe that upon chemotherapy there was no rescue of this vascular damage. This is the case in most of our patients, not all of them, but we don't have a big enough cohort to draw conclusions on that. So this is the reason why we decided to start a clinical trial in collaboration with John Gribben at Bart's Hospital to apply the same parameters that now we set up in mice to patients, AML patients versus healthy controls, before and after chemotherapy. And so the aim of this trial will be to try and understand whether vascular parameters have a diagnostic or prognostic tool in AML and can help directing for the right treatment. Okay, so moving to other models, clearly, ideally, if we want to study the niche, we want to move to the human niche. So this is the reason why organoids models of the bone marrow have been introduced into the field. So the first one I'm describing you is what you were probably already aware of, because it's a paper we published a few years ago in Dominique's laboratory, and is an organoid system that is based on the injection of stromal cells from the marrow, MSCs, and with or without endothelial cells, within sponge scaffolds, together with hematopoietic normal or leukemic cells, and implanted subcutaneously in immunodeficient mice. So we could observe that these organoids are growing very well in 8 to 12 weeks, and they are forming a bona fide human bone marrow. So you can see some pictures here, because we can not only isolate cells, but we can also see cells within these organoids with the intravital 2-photon imaging, and we can study properties such as leukemia motility and organization within the tissue, but also we can study properties of the niche itself, like hypoxia, for instance, measurement of metabolites, or perfusion, as we show in this picture. So this is a very, very powerful tool that we are continuing using now in the lab to validate specific niche targets in the human leukemic microenvironment. Okay, so the last frontier of preclinical model is just trying to avoid using mice and having a system that is as simple as possible but representative of the human bone marrow niche. So try and bioengineer a human bone marrow vascularized niche is a very challenging process, and for several reasons. First of all, because most of the materials that are used in vitro to form organ-on-chips are very different from the real tissue morphology that is present in the marrow, and the morphology of the matrix is fundamental for driving the vascular formation. Also, there is very little direction that is done when these organoids are formed on the vascular formation. This means that vessels are formed as self-organized structure, and this means that they are very different probably from the original tissue. So for that, in the lab, we are trying and reproduce with 3D bioprinting a bone marrow vascular niche unit, in which we can specifically print stromal materials associated to extracellular matrix, bone matrix, and vascular bed, and associate to them the specific cells that are related to arterioles or sinusoids or bone marrow parenchyma and the bone surface. So this is a project that we have just started, and the aim is obtaining a flexible tool to model in vitro the dynamic interaction between leukemia and the bone marrow niche. So, okay, with that I would like to finish, thanking all the people that recently joined the lab, and we have started all the projects. We are really excited working after the COVID lockdown. Big thank also to the Hematopoietic Stem Cell Library at Francis Crick Institute and Dominique Bonnet, and all the other groups that I'm mentioning in this slide, which are collaborators and people supporting my new lab at the Cochin Institute, and the funding body, of course. Thank you so much.